Hi everybody. This is the recording for DNA Technologies lecture, which will be also in a few parts to accommodate the 15-minute YouTube format. Today I'm hoping to talk about Chapter 7, DNA Technologies. I will specifically focus on just these three topics, which is polymerase chain reaction, Sanger sequencing, and then Illumina sequencing. Chapter 7 covers a lot more other DNA technologies, and we will revisit them later when we talk about RNA and protein translation. So it's important to understand that molecular biology is not a theoretical discipline, even though we cover a lot of theory of how various DNA, RNA, and protein processes work. But the power of molecular biology is, in fact, that scientists have, since the discovery of various DNA molecular biology processes, scientists have exploited the various proteins in order to create technologies such as polymerase chain reaction, PCR, which allows us to much more easily manipulate DNA for our own purposes, as opposed to just merely understanding how this is done inside cells by proteins. Before we go into polymerase chain reaction, I wanted to revisit the previous technologies we have already covered in this class, such as DNA extraction. This is just a slide you guys have seen before, so you're on the hook to know how to extract DNA, such as also gel electrophoresis, because this is an important technique, basic technique that's used in a lot of other molecular biology applications. And also we have covered in a few lectures ago when I was talking about RNA structure, the fact that there's two very basic ways to detect specific DNA or RNA sequences not using PCR or sequencing, but using DNA or RNA probes, which are fairly long stretches of DNA or RNA that have either a radioactive or a color label incorporated into them. And because single-stranded DNA will hybridize to matching other single-stranded DNA after being separated by boiling, this hybridization approach has been used for a very long time in order to bind the DNA or RNA specific probes to a target DNA or RNA of choice, because the probe can be detected in some way, whether radioactively or colorimetrically. You can find the specific, whether a certain tissue is expressing a certain DNA, or whether a certain genome contains a certain DNA, or a certain tissue is expressing a certain RNA. Because remember, every single one of our cells contains the same DNA. So I had a mistake here, or not a mistake, but an inconsistency when I last talked about it. So I wanted to correct it, saying that this, the y-axis in this case, is the RNA probes for 3 different proteins, not the mRNAs. And these lanes along the x-axis are different RNA extracted from different tissues that have been run out on, in this case, 3 different gels, and 4 different gels, this gel, this gel, this gel, and this gel, and hybridized to a nitrocellulose membranes in case of these 3, and just the gels were just photographed in this case. And then specific probes for either par delta, par alpha, or par gamma were bound using hybridization at 55 degrees, or whatever the probe likes. To then detect a particular DNA sequence. So you guys can see here that the DNA or RNA has been presented in these two pictures as bands. If you were just to load whole total RNA or whole DNA, genomic DNA, onto a gel and run it, it would not come out as bands. So actually one thing that you always do when you do a southern or northern blot, specifically a southern blot actually, is you cut all of your genomic DNA using enzymes. so that you have many different sized DNAs. You use a common enzyme that cuts fairly frequently, so that you do get this banding pattern on your gel. The genomic DNA has been cut, and then one of these bands will then come up when the probe has bound to it. So I hope this clarified it. And again, there's a lot of videos on YouTube that you guys can look at to better understand how either southern or northern blot works. And please email me questions, obviously, if you have any more specific questions about the topic. Now, because we have already kind of covered 3 different techniques in various lectures, and we will cover a lot more, I highly recommend for people who want to keep all these techniques straight to start keeping a table in Excel, or however you like to do it, so that you can write down what the name of the different techniques are, what the purpose of the technique is, what are the basic materials that you need to use, what is the basic approach or protocol, and what do you think the limitations of these different techniques are. What are their strengths and what are their limitations? So just to give you an example, I summarized these 3 molecular biology techniques that I just reviewed, so DNA extraction, DNA gel electrophoresis, and southern blot, on a table like that. So hopefully this gives you a good idea how to structure all of this information so that you can memorize it, and not just memorize, but have some hope of applying it later in the future. Now, let's go back to DNA replication and the wonderful enzyme that carries out DNA replication, the actual DNA polymerization reaction, which is DNA polymerase. Now, as you guys can see, this is the trombone model of the eukaryotic replisome from yeast. We talked about it last lecture. And here are the 2 DNA polymerases that are involved in the process. Polymerase epsilon for the leading strand and polymerase delta for the lagging strand. So DNA polymerase was discovered originally by Arthur Kornberg's lab in Stanford, and he received a Nobel Prize for it. And after that discovery, DNA polymerase from E. coli was purified, and to demonstrate how DNA polymerase works, the Kornberg group was able to replicate the DNA polymerization reaction in a test tube. So they figured out every component that the DNA polymerase needed. This realization that you don't need to have cells to be able to make more DNA, but you can just use a purified DNA polymerase enzyme and the other ingredients that it needs in order to make DNA in vitro, was the first step in trying to figure out how to develop this very powerful molecular biology technique, which is polymerase chain reaction. Also, I would really appreciate you guys sending me an email saying whether my coverage of the polymerase chain reaction is something that you have already heard and you would like to go into more depth, or if this is something that was challenging enough. Because I know PCR has been mentioned many times in various biological courses, but I don't know in what detail it is mentioned. So I'd really appreciate feedback. Okay, so what are the ingredients for making DNA in a test tube? The Kornberg lab in Stanford, back in the, I think it was actually 60s, realized that you need several ingredients. So first of all, you need purified DNA polymerase, as well as a particular buffer that DNA polymerase likes. Different DNA polymerases prefer different salt concentrations and slightly different pH. And as well, we need magnesium, and I hope you guys remember why DNA polymerase needs magnesium. Magnesium is the critical ingredient in the active site of the DNA polymerase. It is the molecule, magnesium is the molecule that deprotonates the 3' OH group, allowing for the deoxytrinucleotide, triphosphate nucleotides to then be added to the growing DNA chain. So various magnesium concentrations are key to getting a more or less efficient DNA polymerization reaction. And in fact, when buffers are shipped, you frequently have an additional, most DNA polymerase buffers will contain magnesium already, and then there will be an additional tube with magnesium chloride, so you can increase the magnesium concentrations in your buffer and hopefully get a better polymerization reaction. Because the enzyme has to have magnesium in order to catalyze DNA polymerization. So, DNA polymerase buffer and magnesium. You also need a template because DNA polymerase does not synthesize DNA de novo. It needs to read from a single-stranded DNA molecule, not double-stranded, has to be single-stranded. Now, when DNA replication occurs in a eukaryotic or prokaryotic cell, the double-stranded DNA helix is specifically unwound by helicase. In the case of a DNA polymerization reaction in vitro, it's done differently. There is no helicase added in this case. You also need a DNA primer that will bind to your DNA template and provide the DNA polymerase a binding area from which it can elongate the new DNA strand. It has to have an intact 3' OH group. That's very important because the 3' OH is critical for the DNA polymerization reaction. And of course, you need the material to polymerize DNA from, so you need nucleotide triphosphates. In case of making DNA for PCR and so forth, you need deoxynucleotides, so that's D-NTPs. You need all 4 of them. And this is basically all the members coming together. There's the primer that has bound or annealed, glued itself to the template strand in orange. There's your DNA polymerase, and then there's this phosphate, nucleotide triphosphate molecule coming in, ready to be polymerized to the primer and allow DNA polymerase to extend the chain. This reaction will proceed in a test tube without any additional energy. You don't need to add ATP in order for DNA polymerase to do its job. You have to have magnesium. And the reason you don't need to add ATP is because you already have added DATP, DTTP, DGTP, and DCTP, which are energy-carrying molecules with three phosphate groups in the end, and the phosphate groups are unstable, so as the chemical energy released during the reaction will allow for the polymerization to occur. So let's say you would like to, you found this beautiful green glowing coral that glows under UV light, this bright green or bright red or bright orange, and you would like to figure out what sequence of this gene is. Now, let's say you have extracted DNA from a coral and now you end up with a bunch of these chromosomes. How do you tell the polymerase where to start? Now, from the previous slide, you remember that DNA polymerase needs a primer, but it doesn't have to be just any primer. If you want to isolate something specific from the vastness of the coral genome, which is 420 million base pairs, you need to give the DNA polymerase a specific primer that will bind in the 5' region of the gene that you're interested in isolating. So the sequence will hybridize to a complementary sequence inside the coral DNA. So for example, if you have a primer that's CCTA, it will anneal to the sequence inside the genome, and it will be the, if you just look at the base pairing here, you will right now see it as GGAT, but what you actually would be looking for if this was your template and this was your primer, you would be sending the primer sequence TAGG to your primer sequencing company. And why? I hope you guys remember that DNA, if in doubt, realize that DNA is written 5' to 3'. Now, I try very hard throughout the course to mark what the 5' and 3' end is, but when you're dealing with a sequencing company, they just assume that what you're giving them is 5' to 3'. They will synthesize the DNA in the reverse orientation, if you guys remember, that's the technology I forgot to include. If you guys remember from the DNA synthesis reactions, the chemical synthesis actually goes 3' to 5'. But you need to submit your primer sequence 5' to 3'. Now, let's go back to how we tell the polymerase where to start. And I said, add a specific primer. This primer will be specific against the sequence of your fluorescent coral fluorescent molecule protein. But there's a problem here. Again, the coral genome is about 4 times 10 to the 8th base pairs. How do you make sure that your primer will specifically bind in only one location? You need to make a primer that's long enough. A 4-nucleotide primer is not good. It's not good enough to specifically select one particular sequence out of 420 million base pairs of a coral genome. So if you would like to calculate the probabilities, that's basically the formula. There's 4 probabilities of what can be in a given location inside the DNA sequence, because there's 4 different nucleotides. And the nth power is the length of the primer. How many ways, this formula will give you the number of possible arrangements of a sequence of a certain length. So if you have a primer that's 4 nucleotide long, that's sufficient binding for the DNA polymerase to begin extending from it. But it will only give you 256 different probabilities, and so if you have a genome with 4 to the 10 to the 8th base pairs, this 4-nucleotide sequence will appear very, very frequently, and so your primer will not bind specifically in one particular location, but it will bind in many locations. And amplify not just the DNA from the fluorescent gene that you're interested in, but all kinds of other stuff. Now, if you take a nucleotide that's about 18 when you take a primer that's about 18 nucleotides long, you get about 7 to the 10 to the 10th possibilities, which absolutely covers the 420 million base pairs of the coral genome, assuring that your primer will bind all 18 base pairs exactly once. However, there's still a potential that your primer will base pair imprecisely to other locations. So whenever you do a polymerase chain reaction, be aware that even though you've designed a specific primer and it's 25 nucleotides long, and there shouldn't be a mistake, you will still get additional bands on your gel, and you will need to play with concentrations of the primer, the template, the magnesium, or the annealing temperature, which we'll discuss in a second, in order to make sure to either optimize the reaction to only get one product, or you can also cut out the band out of the gel when you run it out. So this was a little bit too technical for beginning, but hopefully you guys appreciate from the slide that the sufficiently long specific primer needs to be designed in order to assure that the DNA polymerase amplifies only one sequence from the template as opposed to many sequences. Now, let's go back to our goal to copy the fluorescent protein from the coral genome. So let's say we did the step 1, so we extracted genomic DNA from the coral. Now, since DNA polymerase needs a single-stranded DNA molecule, and we don't add a helicase, it will create a single-stranded molecule from a double helix. Instead, people resort to the brutal means of just heating the DNA molecule and to the near boiling, so that it denatures. All the hydrogen bonds between the base pairs come apart, and instead of a double helix, at the boiling point, you end up with 2 single-stranded stretches of DNA. Now the second step, then the third step is to add a specific primer for your specific gene that you're interested in. So let's say you added a bunch of primers to your reaction. At the near boiling point, there's no possibility of forming hydrogen bonds. That is why your DNA molecules have melted apart. But if you cool the mixture slightly to, say, 60 degrees, that's still not enough. There's still a lot of thermal motion for these big molecules, and they haven't fully base paired yet. But the little primers, which only need between 18 and 25 base pairs in order to form a double-stranded helix with the template, will have bound. They will have hybridized to their target sequence, while the big strands are still not fully annealed. And now that you have gotten your primer to form a double-stranded hydrogen bond setup with your template, you can now add your DNA polymerase and your nucleotides and your magnesium, and hopefully your buffer was there already, in order to allow the DNA polymerase to bind to the primer and then extend this muddy green sequence of the newly synthesized DNA strand. And here we go, congratulations. You have just made one copy of the coral gene. Now you have the blue copy and you have the green copy. And these three steps of the typical DNA polymerase, the need to denature DNA, the need to anneal the primers, and the need to elongate with DNA polymerase are the steps of a typical PCR cycle. So template DNA is usually denatured at 95. Primers are annealed at various melting temperatures of the primer, depending on the length of your primer and the GC content of the primer, and there's a lot of softwares out there to help you calculate it. So between 50 and 72. And then you need to elongate with your DNA polymerase. Now here comes another problem. At even 72 degrees Celsius, most proteins, including DNA polymerases from bacteria such as E. coli, will have denatured, or at least partially denatured. They will have definitely denatured at 95 degrees. That is required for denaturing, for separating the two strands of a DNA helix to serve as template. Because the tertiary structure, the three-dimensional structure of the DNA polymerase is critical for its function. Otherwise, the aspartate residues are not in the active site. They're not creating this nice little pocket for the aspartate residues to hold the magnesium in just the exact location, in just a precise orientation, so the DNA polymerization can occur. So at 37 degrees, a DNA polymerase from E. coli will be functional, but at 95 it will not be. It will be just a strand of amino acids completely denatured. So how do you in the very, very early stages of DNA polymerization experiments in vitro, the scientists would add DNA polymerase after every cycle of this polymerization reaction. But if we could have a DNA polymerase that is stable at 95 degrees and doesn't denature, wouldn't that be great? Well, not all life requires 37 degrees to live, to function, and there is some extremophile bacteria and archaea out there. So basically at the same time and slightly earlier than when the PCR reaction protocol was created, scientists from Minnesota isolated a very heat-stable bacteria, the thermophilic bacterium Thermophilus aquaticus, from one of the hot springs in Yellowstone National Park. So it's the mushroom pool, which is not one of the famous ones. It's sort of another location from the big collection of the prismatic pool and so forth. And again, because it's not as hot as a prismatic pool, doesn't have quite so colorful of the bacterial population growing them, so it's a bit brown. So, however, the scientists were able to isolate this thermophilic bacterium out of the hot waters of the mushroom pool. And then they were able, using approaches we'll discuss later called degenerate, they were able to isolate the sequence for the DNA polymerase from the bacterium. And because the organism is called Thermophilus aquaticus, the polymerase isolated from this bacterium is called Taq polymerase. It's a thermostable DNA polymerase. Its optimal functional its optimal polymerization temperatures, when it's very effective at doing its reaction, is the temperature of the mushroom pool from which it was isolated, which peaks, the polymerization reaction peaks at around 72 Celsius. So that is the step described here. And it also is not fully denatured at 95. It holds its shape really darn well, allowing for repeating of this 1-2-3 cycle of a typical PCR process without the need of adding additional enzyme. So you guys can see here the DNA polymerase, the Taq DNA polymerase works at both high temperatures, that works at high temperatures that would destroy the polymerases from any other bacterium that grows at 37. It's fully functional, very, very fast at 72, and it doesn't fully denature at 95 and can easily revert to 72. Now, so let's go back to our goal of copying a gene from for fluorescent protein from the coral genome. I kind of changed the steps here. So instead of step 1, this is now step -2. Because I wanted to highlight the 3 steps of the PCR reaction again and again. So first we extracted DNA from our coral, and we figured out what the specific primers are for that will bind the DNA region that codes for the fluorescent protein, and we added the specific primers, the DNA template, the genomic DNA, and the Taq polymerase. And again, now we're going to heat to melt the DNA helix of the template. We will cool the reaction to just enough for the specific primers to hybridize to the target sequence. And then we will warm up the DNA polymerase to allow it to extend at 72. Now, why don't we keep this at 50? Why don't we worry that this primer is going to come off? Well, as soon as the DNA polymerase binds the primer, it will start elongating the chain, creating many more base pairs. So if the primer denatures off of it, that's still okay. It's the DNA polymerase is optimally functional. The Taq DNA polymerase is optimally functional at 72. So that's the temperature, that's the preferred temperature to keep your DNA, to keep your reaction at, to allow DNA polymerase to extend to its best ability. So depending on the length of this step 3, the elongation step, the DNA polymerase will be able to extend a few hundred nucleotides, a few thousand nucleotides. And the ballpark estimate for Taq DNA polymerase, as opposed to the more advanced ones, is about 1,000 base pairs a minute. So if your product is about 1,000 base pairs, your elongation step will be 1 minute. If you're trying to make something that's 8,000 base pairs, you would be waiting for 8 minutes for the DNA polymerase to do its job, if you want the full 8,000 nucleotides. And then, because DNA polymerase from Thermophilus aquaticus does not denature, you can continue repeating this cycle. You can go step 1 to 3 and back, step 1 to 3 and back, step 1 to 3. And the more steps you make, the more of the DNA copies of your gene of interest you will have amplified. So instead of making 1 copy, you have now made some 35 copies after 35 cycles. But one can do it even better. And the important step in optimizing and developing the actual polymerase chain reaction came from the realization that if you use 2 flanking primers, instead of 1 primer that the Kornberg group was using, you can get more than 1 copy of DNA per cycle, With one primer, you only get linear amplification of the product, so one cycle, one extra copy. While with two primers, you'll get an exponential amplification. So here we go. At 25 cycles, the two primers situation will produce about 1 million copies of your gene. The other realization came is that if you use two primers, you can really define the area that the DNA polymerase will amplify and produce not a bunch of fragments of different size depending on when the DNA polymerase fell off the template. Sure, you give it 8 minutes to get 8,000 base pairs, or 5 minutes to get 5,000 base pairs, but not every DNA polymerase will stay there until the end. Sometimes they fall off early. Because remember, the DNA polymerase that we're using here does not have this clamp that clamps it onto the template. It's just hanging on by its own DNA binding ability. So you guys can see here that there's the orange are the primers, and then after a while you get a bunch of fragments of the short size. There'll still be some molecules that are template DNA. It doesn't just magically disappear. But the overwhelming majority of the molecules will all be the same length. And all of this idea of using two flanking primers, and also using the idea of using a thermostable DNA polymerase tag, which was discovered by other people, but all of this was put together by Kerry Mullis, who was a research scientist for this CITES company in San Francisco. And he's probably one of the goofiest people to receive the Nobel Prize. This is the book and the book cover that kind of exemplifies what Kerry Mullis is all about. Now, the PCR reaction protocol was published in 1986, and then Kerry Mullis and others received the Nobel Prize in 1993. So it took them a while before they were willing to give the prize for PCR. And I bet there's a bunch of people here right now listening to me that were born after 1986. Time flies.